So yeah, as Robin already said, what I'd like to show you today is a bit what kind of work we do in our lab in Copenhagen, in Denmark. We are a very recent lab, a very young group. So in the current formation, we are existing for about a year. So what I'd like to talk to you about is not so much about specific research results, but more about what our vision is and what we plan to do in the future. So we are based in Denmark at the Statens Serum Institute, which is basically the Danish Public Health Institute. So our institute, among others, provides different health-related services to the public domain, such as vaccination, the screening of newborns, but also more recently, it hosts the whole COVID test center. But also, we do have a very active research environment, and also we host very big national centers, such as Danish National Biobank, as well as the Danish National Genome Center. A bit more about those institutions just now. So the Danish National Biobank hosts a total of 8.5 million samples in store, and with that is one of the biggest and most advanced biobanks in the world. And so there is also a very unique system in Denmark with which we can link all of those biological samples to Danish register data. So this Danish register data contains information about both socioeconomic aspects of the person, but also health records about diseases and different pathologies a person went through during their life. And so this is, of course, a very unique resource to do population-based research. And so within the Statens Serum Institute, our group is based within the section for clinical mass spectrometry and the Danish Center for Neonatal Screening. So the routine part of our department, they regularly receive all dried heel prick samples that they screen for a number of metabolic diseases. So basically every time a baby is born in Denmark, a heel prick sample is taken, and those samples are then stored in the National Biobank, where they currently sum up to over 2 million samples. So that's about half of the Danish population. And so the objectives of our research group is basically to use these samples, this massive population-based resource, to study early predictors of disease using metabolomics methods. And so our vision is basically that sometime in the probably more distant future, we will have metabolomics methods that are so good that we basically can predict disease at birth, fostering predictive, preventive, and personalized diagnostics. And so we think that metabolomics is really an ideal tool to develop such methods, because the metabolome, as you know, reflects many key factors of our health. So it's all small molecules that our body synthesizes, metabolizes, comes in contact with, or also ingests. And so together with various collaboration partners, we investigate different research questions and research projects. So we have projects that are more broad scientific questions, such as how do pregnancy exposures shape the neonatal metabolome, or what defines the metabolome of prematurely born children. But we also have more applied research questions, such as are there metabolic predictors of mental disorders at birth, or can we predict central nervous system tumors at birth, or also are there any metabolic markers of inflammatory bowel disease at birth. Then we also have more methodological studies, such as, for example, how does the storage time affect the dried blood spot metabolome, because those samples are all stored for many years, so they are collected since the 1980s. So of course, it's very important for us to know, are those metabolites still stable, and can we still use them to make any meaningful conclusions in our research questions. And also we have more like computational methods-focused project. So we work with very huge sample sizes. So typically a couple of 100 to 1,000 samples, and many of the processing algorithms that are out there are at the slower range. So we are getting to the limits of the power. So we also work with developing automated methods for metabolomics data preprocessing. And so what are our analytical and computational tools? We have currently two mass specs. We have a Bulk Impact and also a Bulk timsTOF. And then for all our data processing, because our data is considered person sensitive, so we are unfortunately allowed to share the data in the public domain, but we are using a supercomputer, the National Genome Center, which also hosts patient data from the whole of Denmark, including genomic data, and they have a very special secured system where we can work with our data. And so basically our mission is both to develop analytical as well as computational methods that we can establish cutting-edge mass spec methods for the better understanding of disease etiology and diagnosis, but also to develop fast computational solutions that we can analyze those population size metabolomics data sets. And the reason we are doing all of this, of course, is we want to have a translational aspect. So our goal is that we can discover some biomarkers that can be implemented in the clinic, for example, in the neonatal screening. And so we can build recommendation systems for doctors. And so just to mention, like all we are many tools that we use on the computational side are tools that are implemented within GNPS. So it includes molecular networking, but also many of the great tools, some of which we have discussed at the summer school. And really our goal is that we can pinpoint known as well as novel biomarkers of disease. And so one question I get often from the clinical work mainly is, do we even have any new metabolites in human samples? Is it not just better that we do a targeted approach and then just measure the metabolites that basically are always there in the neonatal dried blood spots or in plasma in general? And so I think the work of a colleague of ours in our former lab, in Peter Dorstein's lab, is a very nice example to contradict this. So in his study, basically the background is that we have bile acids, and we know that there are primary bile acids. We know that they get dehydroxylated by bacteria in the gut, and they form secondary bile acids, which can be conjugated with glycine or with taurine. And this is a bit of a provocative statement, but more or less that's what we knew for many years of research. But with molecular networking, actually, Rob Quinn and his collaborators found that there's also bile acids that are conjugated to phenylalanine, to tyrosine, and to leucine. And so also some of those were linked to disorders such as inflammatory bowel disease or cystic fibrosis. I think this just very nicely highlights that it's very relevant to find also new metabolites in human samples, and also nicely highlights really how powerful those tools can be that we learned about in the summer school. Then just to mention some more specific research output we have created in the past year. So one basic question we asked is, can actually the neonatal metabolome predict health? That would be very important if we want to continue any of our research. So what we used here is a deeply phenotyped cohort from the Copenhagen Perspective Studies on asthma in childhood, mother-child cohort, and basically using a ridge regression, we could find that based on the metabolome, we can predict the number of days a baby is hospitalized. So this shows nicely, I think, that there is something in the metabolome that will predict disease states or will give us a clue about how well a person is. Also, we were interested what are other key determinants that shape the neonatal metabolome. And so again, we used this deeply phenotyped Copsack cohort where we had a very rich source of phenotype data, 21 in total. So about how the baby was born, natural or a C-section, clinical data, hospitalization, also at what season of the year it was born, and so on. And so there we used light GBM graded boosted decision trees, and we could find that almost 50% of the metabolic features that we found in our babies, they can be predicted by pregnancy or early life exposures, like highlighting that it's the time during pregnancy and in early life is very important to shape the neonatal metabolome. So one of the very strong effects we found was the seasonal effects. So like funnily and very interestingly, babies born in winter have a very different metabolome than babies born in summer. We don't know the exact reason for this yet. It could also have to do with transport or storage time, but more an observational data anecdote we found is that, for example, citrus biomarkers are predominantly found in children and that are born during the winter months. So we have here a different citrus metabolites highlighted in this molecular family. And also we found some pesticides, and we found they are slightly more abundant in children that are born in summer. We could just speculate, maybe that's because the mothers eat more fruits, or yeah, there could also be other reasons, I guess, for that. And also we can detect many metabolic markers of maternal exposures in those neonatal bright blood spots. So that reaches from medication to antibiotics to cotinine, which is a marker for smoking, but also different food biomarkers, pesticides, as well as some microbial metabolites. And so another interesting factor we observed is that the child's age has a very strong influence on the neonatal metabolome, so gestational age, and that is with how many weeks in the belly the baby is born. And so we see here this nice gradient from children in yellow that are born very late during gestation, and children that are born very early during gestation, so premature babies. And using many of the tools we discussed during the summer school, we could identify some of the metabolites that are related to gestational age, such as bile acids, carnitines, nucleotides, polyamines, but also care treatment and treatment-related metabolites, and also some of the microbial metabolites. And so some of those metabolites we detected that might be from the gut microbiome. They were also associated with some of the adverse outcomes of premature birth. So there could be something behind that we can use this metabolomics data to better understand the disease etiology in that case. And then we also have a study that was led by Philip on how storage time affects the bright blood spot metabolome. And Philip will now present some of his findings to you by himself.
Thank you very much. Yes, so in order to be able to use all these samples that may have been stored in freezers for years or sometimes decades or longer, we of course need to make sure that they are high quality. So in order to investigate this, we designed this study where we retrieved 200 dried blood spot samples from the Danish Natural Biobank that originally were collected throughout the time period of 10 years. So 20 samples per year, And the idea is that we wanted to build kind of a temporal trend, so to see how the levels of this metabolite varied over time, depending on the storage. So of course, these samples were taken from different babies originally. So there will be some biological variation that we cannot really control for perfectly, but we try to take this into account as much as we could in order to select samples that minimizes this known variation that we have observed in previous studies. So for example, we wanted to select samples that were both from male and female babies. And we also sampled children that all were born in July at the same hospital. And they were all sampled at exactly 2 days of age and 40 weeks of gestational age in order to minimize these biological variations. Could also adjust for in statistical analysis later on for factors such as birth weight and the mother's age to make sure that to minimize this variation as much as possible. Anyways, so this ended up being 200 dry blood spots. We analyzed them using our LC-Q-TOF system. We did a pre-processing using MZ-MINE, ended up with 731 metabolite features that we annotated using GMPS and series. Ended up with around 180 annotated metabolites, while the rest were unknown features. What we found is that just using a normal linear regression model, we found that around 80% of the metabolite features in our data sets were stable during these 10 years of storage. We tried to then also adjust for factors such as sex, birth weight, and the mother's age in order to minimize the variation from the samples. Of course, there were some metabolites that were quite affected by storage, such as methionine and glutathione, and they were also like dispersed throughout like a quite wide variety of different metabolite classes. But we see this as a good way to kind of keep track of which metabolite we may have to look more into in future, whether we can trust data that is comes from samples that may have been stored for several decades. If we just look at a little bit how the levels changed over time, we see that it was more like expected, I guess, a gradual shift in the metabolite levels over time. So the positive are the ones that increased over time, and the negatives the one that decreased. So you can see that it's more or less like a gradual shift throughout these years of storage, which I guess is kind of expected. We also wanted to see if we could explore a little bit potential ways that these metabolites were transformed during storage. So in order to do that, we used mass spectral molecular networks. And we set up 3 criteria in order to define a potential transformation pathway. And the first is that the metabolites were themselves associated with storage time. The second, that they correlate negatively with each other. And third, that they have a mass spectral similarity, so a cosine score above 0.7, or that they share a MS2LDA substructural motif. And while doing that, we found one potential transformation pathway, and this was for glutathione, where we could see that both glutathione and the crotonyl aldehyde conjugated version of glutathione were decreasing over time, and they were negatively correlated with an unknown glutathione isomer. And they shared either MS2LDA motifs or mass spectral similarity. And you can see also the temporal trends on the graph to the right. You can see that the glutathione is decreasing while this other unknown isomer is increasing over time, more or less to get some idea of what's going on. But in overall, we found that at least that the majority of the metabolite seemed to be quite stable, and we more or less generate a list of metabolites that we want to keep track of in the future. All right, so over again to you, Madeleine.
Thanks a lot, Philip. So yeah, with that we have come with that, we have already come to the end of our presentation. So this is basically us. You can see we're almost everyone is here at the summer school. Francesco and Philip and myself. We have Nadia as well. She's at home in Copenhagen. She's our lab tech. And so we would like to thank all our collaborators, of course, as well. And also we hope that we could interest you a bit in the types of data we work with. We are always super open to collaboration. So if you have ideas for projects or ideas you want to develop some algorithms with our data or have other projects ideas for collaboration, we are very open to that. So feel free to always contact us about this. And also, hopefully we will soon be hiring 2 computational metabolomics academics to join our team. So if this interests you, please either contact me directly or keep yourself posted on LinkedIn and Twitter, where I certainly will post those positions. They are not online yet, but this will happen in the very near future, hopefully. And yeah, thank you very much.